This morning, it's going to be my pleasure to introduce our two speakers this morning. We're going to hear from them sort of in a simultaneous fashion, one right after the other. Dr. John Rizzo, J.R. Rizzo, is one of our own. He is a member of the CRF. He sits on the CRF Board of Directors, but perhaps even more importantly, he's a true physician scientist and holds a faculty appointment in several departments at the NYU Langone Medical Center. He's a faculty member of the Department of Physical Medicine and Rehabilitation with a cross appointment in the Department of Neurology and Departments of Biomedical and Mechanical and Aerospace Engineering at NYU. He is also the Associate Director of Healthcare for NYU Wireless Laboratory in the Department of Electrical and Computer Engineering at the Tandon School of Engineering on the NYU campus. He's the founder and chief medical advisor of Tactile Navigation Tools, where he and his team work to disrupt the assistive technology space for those with visual impairments of all kinds. So really, he's trying to enhance human capability. And again, as I mentioned, he sits on our board of directors of the CRF, and he has also had a webinar that he's given out, and you can see that on YouTube if you're interested. Also this morning, we're going to hear from Dr. David Gamm. In the world of academic medicine, you have individuals that are called triple threats, and that's an individual who's an outstanding clinician, researcher, and also an educator, and Dr. Gamm clearly is that. He's a professor of ophthalmology at the University of Wisconsin-Madison, and a professor of both ophthalmology and visual sciences. He's the director of the McPherson Eye Research Institute and member of the Weizmann Center Stem Cell Research Program, and the University of Wisconsin Stem Cell and Regenerative Medicine Center, and the American Society for Clinical Investigation. His research efforts have been directed mostly toward basic and translational retinal stem cell research, and he and his team have been working with us and our organization for a number of years. And I've gotten to know him a little bit better over the last intervening year and have been amazed at his ability to tolerate all of my pestering questions and also take a very impressive repertoire of scientific understanding and be able to present it to me in a way that I understand. Also importantly, and he is here today with Kim Edwards. Kim is the very first recipient of the Randy Wheelock Award, and we're grateful for her presence here, and we're also grateful for his mentorship of her. So without further ado, I will present to you Drs. Gamm and Rizzo, and they will take it away. I think we're going to start with Dr. Rizzo, and then we will have some Q&A between and then at the end as well. I believe. Yeah, whichever you prefer. I think just open it up. Yeah.
Sorry. Yeah, sure, I could say. So good morning, everyone. It's a pleasure to be here. As a CHMer myself, I really have to give a strong shout out to all the leadership at CRF. The CRF's been an inspiration to me, really since its inception, and I really can't thank you all enough for all of your work over the years. So it's really a pleasure to be here and also to speak alongside Dr. David Gamm. So really happy to be here. Just one kind of housekeeping announcement. I'm going to try and use, as you're facing the stage, the right screen when I go into my talk. So if people want to slowly migrate over to the right side, I do have a special laser pointer, and it's kind of low vision enabled, and I'm going to try and use that to walk you through a lot of visual content if possible and narrate that experience. So I just wanted to give that as a heads up. But we wanted to talk a little bit about both fundamental treatment and also functional treatment. So maybe I'll pass the microphone back over to Dr. Gamm, and then we'll chat with you for a few moments before we get into our actual talks. Yes.
So good morning. I'm David Gamm, and all the superlatives I think were greatly exaggerated. So I always like to have a low bar to start with. So, but it's great to be here. I've been involved with CRF for a very long time. After starting with when, and I'll mention it a little bit later in my talk, but when Randy Wheelock grabbed my elbow at a meeting and implored me to start working on it, which was not hard to do since it was certainly something that we had a great deal of interest in, and that I had seen affect patients of mine at UW-Madison in my clinic. So it's great to be here, and we have a lot of ground to cover, I think. And so in this first 15 minutes, we were charged with talking about how technology has impacted CRF, and that's small subject, I suppose, too. And but so I thought that I was talking to JR earlier, and that maybe we'd break that down a little bit, and we want to have enough time to answer questions at the end too. But I'll just start by saying, kind of, this is all going to be 1,000-foot stuff, but really when it comes down to how technology can help choroideremia and families with choroideremia dealing with it, it starts with basic science research and just how well we understand the disease. There's so much we still need to better understand in terms of what cell types are being primarily affected. What is it that the gene and the protein product are doing inside of the cells? Why is it that it seems to be uniquely in humans, so on and so forth. And by understanding how something works or doesn't work, that helps us to refine how to fix it. So just like you can use all the analogies you could think of, if you wanted to fix a car or you wanted to fix your sink, if you didn't know going into it exactly what was wrong, it would make it hard. So the technology from a laboratory standpoint, just to be able to look at the basic mechanisms of disease, have skyrocketed. So over the last 20, 30 years, the types of things we can do to better understand the molecular biology, the cell biology that's going on, has been very assistive. And then from there, then you have translational research, so moving that into animal models as that bridge to clinical trials, and then technology for us to be able to understand what's happening in human patients via imaging and some of the clinical tools that we have. So all those things kind of come together to help patients of all types, but in particular, I think choroideremia, because in many ways the eye is leading the way. So there's a lot of things that are beneficial to working in the eye, not the least of which is you have two of them, so you can mess around with one and leave the other one by itself. But it's also accessible, right? It's not your brain, so we don't have to put you in an expensive scanner to be able to see what's going on. We can dilate you and take a look. There's all sorts of very effective types of imaging, functional imaging, structural imaging that are fairly routine now in people's offices that you walk into. And so all of those things are an advantage when it comes to the eye versus other organs. Plus, it's an organ that is relatively encapsulated, so you can work within a somewhat isolated space, which from a safety standpoint is important. And then there are a lot of things that, a lot of aspects to the cells that we work on that are very unique in the body. And so we can manipulate things specifically within cell types, and we don't have to worry so much that when we do that, it might affect some other cell type in an organ that's perfectly fine in folks with choroideremia, so we'd rather not mess with the stuff that works. So all those things put us in a strong position to be able to advance the field on many different fronts. And so with that, I'll move it back to JR and maybe go back and forth.
Sure. Thanks, David. So, I think that's a fantastic primer on technology. But, when we think about technology, I like to use framework, right? And I would encourage you all, as someone who's trying to imbibe or drink a lot of information constantly, you could think about kind of technology on two sides of a coin, right? You have technology that's trying to advance basic science. And so that's really treating the eye as an organ, as Dr. Gamm was just referring to. And then you could also think about technology that's trying to advance the person. There are different conceptual frameworks that are out there, specifically within the rehabilitation medicine space, which is where I hail from. And you could think about that as vision function versus functional vision, right? So it's really trying to treat the eye as an organ or treating the person. And the reason I think it's important to think about that framework is that when we read about technology, there's all kinds of amazing things happening in both worlds, in both domains, right? And we can really advance a lot of what's happening on the basic science side. which is really helpful for things like gene editing or gene therapy. But then on the other side, when we talk about assistive technologies, we had a great lecture yesterday, and there's all kinds of things happening. Right now, we're in the fourth industrial revolution, and I'll touch on this in my talk shortly. But everything is just moving forward at such an accelerated pace, right? We're going to talk about driverless cars, and there's so much innovation in terms of sensing technology and computing power, and can we actually use that? So, some of our questions going back over a decade has been, all these advances in driverless car, can we apply them to visionless pedestrians, right? And how can I advance the function of a person? So I really think it's very helpful to think about technology kind of in that framework, and with an eye towards those kind of two different domains, if you will. And if you do, I think you'll be doing yourself a good service. I also think when you're speaking with clinicians, it's helpful to understand and to speak about these things with them and to advocate for yourself, right? It's good to be talking about both a fundamental treatment to address any issues at the organ level, but also about what's happening behaviorally with you at the person level. And classically in medical school, when I went to medical school, we don't really talk about these things, right? But it's really important. So I would argue when you get into those scenarios and you're just, kind of simply referred for services to talk about you as a person, you should advocate more strongly to say, hey, listen, I want to understand more about how I could be treated as a person in terms of what other technology exists. What are the other offerings in this space, right? And you can think of this as kind of your fundamental cure addressing that organ, or your functional treatment or your functional cure. And if you think about it in those two ways, every time you see a clinician or you see someone in the medical space, it's important to have that dialogue with them. So you're always on the avant-garde. You're always getting the best of the best. And, I think it's really important. In general, I feel like we're at a disservice. And I'll show you a slide later on in the presentation. If we look at the physical impairment space and the world of assistive technology, I would say they're kind of a leap year ahead of us compared to those on the sensory impairment space, which is really to our disadvantage. Because all of us have serious visual disability, and every time we take a step in the real world, we're at a disadvantage, right? And it would be phenomenal if we had more technology to help take care of us. And unfortunately, the way a lot of these payer systems are geared and the government is currently situated, we don't have strong reimbursement mechanisms for these devices. But we need to talk about it, and we need to advocate for it. And it's important to think about it in these kind of two different ways. So I'll walk you through kind of the different possibilities, but I'll leave you with the fact that, at least in this short little nugget here, that the future is incredibly bright. And especially over the last 5 or 10 years, technology is everywhere and it's advancing so fast. So we need to, really pay attention and try and take advantage, but advocate for it so we're getting the attention in our immediate space. And all the other areas aren't, kind of trying to steal all the limelight, so to say. I'll pass it back over.
So to reinforce what you just said, I tell my students all the time, I say if you're in a battle with engineering, between engineering and biology, engineering will win. Because they're exceedingly smart and they have a lot of things at their fingertips that they can do. And the realms at which engineering is now moving into, from not just quality of life standpoint, but actually kind of melding into what we used to thought was exclusively the realm of biological treatments, that's really getting blurred. So we're seeing a prime example would be people in my space who are interested in making insulin secreting cells from stem cells. Well, the technology for detecting and doing implants that will release insulin, so purely an engineering approach, are getting so good that it would be hard to justify having a cell do that if you can have a device do it just as well, which can be removed and has isn't going to get rejected, things like that. That's why in many respects, I keep a close watch on the prosthetics in the eye, and there's visual cortical prosthetics that are being developed right now for folks who, say, for instance, have blast injuries from military service who don't even have eyes. And so there's all sorts of different, if you look at where your seat of disability lies, whether that be, in a specific cell in the retina, in the optic nerve, in the brain, there's always a way to think about how do we bypass that. Now, in my field, what I try to do is I try to replace things and try to restore the normal order as much as we possibly can. But the ability with engineering and technology to be able to bypass that, I think, is getting exceedingly good and very exciting to watch. From a gene and cell therapy standpoint, so switching over to what I'm more familiar with, over that's exponentially grown in terms of that technology. If you people in this audience, probably a lot of you are very well aware, there was a time not too long ago when your only option, when we talked about gene therapy, it meant one thing. It meant that you were trying to insert a complete copy of a gene that didn't work in a particular patient. And that was limited to the size of that gene, because you had to, how big was the truck you could get to deliver that gene? Would the truck actually get to the cells that need them? And there are all sorts of limitations with regard to that. Now, that's still the primary way to do, quote, gene therapy, but there's dozens of different types of gene therapy now that include trying to fix the gene, trying to fix the go-betweens between gene and protein manufacture called RNA. You can try so there's ways to read through, and I'll talk about a little bit too, different types of mutations. So we've gone from there being, gene therapy being put on hold decades ago because of some untoward effects in the first patients, and then learning through that in Gene Bennett's work, and then ultimately getting to where gene augmentation or gene replacement therapy was back on the map, and now is still the most concentrated area of gene therapy. But I work with companies every time I talk to a new company with a new type of technology, it's exceedingly interesting. There's so many different ways to try to approach different types of genetic disorders. And all of them are interested in having an ocular program. They may not ultimately, that might not be their focus, but they all see the eye as having great advantages to beta testing whatever it is. And they may ultimately want to fix, hemophilia, but they want to start in the eye. And they're looking for model systems and ways in which that they can test the safety and efficacy of their products before moving on to other parts of the body. So in that regard, the eye really still remains to be the tip of the spear on a lot of the technology development, which is good and bad, right? I mean, it's good because you get the first crack at things, but you also have to be careful about safety. So you also always have to remember that alongside of all of the advancements in technology, we have to be cognizant of the fact that they introduce new and unique safety concerns sometimes too. And so our ability to be able to monitor those is just as important as our ability to try to think of a new way to perhaps fix a problem. So, and then from a cell therapy, I saved what I do actually for the last. A lot of advancements there has been on the manufacturing side. So the promise from the very beginning was that human pluripotent stem cells would be a source of authentic cell types. So basically, cells that are damaged or lost in the course of disease, you now have, at least theoretically, a source that you can manufacture them. And initially, that was a great pie-in-the-sky idea, but actually taking those cell types and being able to direct them towards the cells of interest, which sometimes are exceedingly complex, like photoreceptors and RPE, making sure that they're real cells and they don't just look the part. It's a real duck, not a wooden duck. That's taken a lot of time to do, and then to convert that to a method that is compatible with putting back into a human, so that it's safe, you can scale it up, it makes sense to do it. That has advanced tremendously, and to the point where we're really on the precipice of being able, we're essentially doing that right now. Installation, getting those cells, those new cells, that the new wine to go into old wineskins and have those cells be able to connect and talk to the downstream cells that have been sitting around in the face of a lot of disease. I mean, that's an area that still needs work. We and many others are working towards. So that's where I would foresee a lot of the advancements in technology going in the near future. So, hand that back. Although I think we've probably managed to talk enough, haven't we?
I'm not sure. How are we on time, Jess?
Good.
We're good on time?
So maybe do we have time for questions before we go?
Dr. Gamm, we hear about the advancements that are being made in advanced optics, in microperimetry, OCT, new OCTs. Can you explain to the patients the need for potentially getting FDA approval, how and what are the challenges of getting these new advanced ways of measuring, especially for potential endpoints, to have those as possible inclusion in a trial and the complexity of that?
That's a bit of a loaded question.
Yeah, thank you. I thought I'd give
So,
that to you.
because the first thing to remember is that not every technology we have to rush and get in, and have FDA approval and start throwing them into clinical trials. A lot of it, we don't know enough about it to really be able to interpret. They may have, a small cohort of patients. We see some exciting things, but they really haven't been validated yet. I mean, the FDA is all about validation. Now, they can be very slow to do that. You also have to pick and choose what you want to use in a clinical trial as an endpoint because it's expensive. You can't just have 24 measurements because each one of those has a price tag to it. So it is probably a slower process than we hope that it would be. And there are some things that the FDA still hold on to that make it difficult to ultimately bring products to market. So for instance, they have a very high bar for visual acuity for them to be able to say, yeah, that product really helps people. And so for a long time, they needed to see an improvement in 3 lines of vision. Well, that's hard to do. That's very hard to do. That doesn't mean that a patient who gets that therapeutic isn't greatly benefited in their activities of daily life, that their vision isn't improved. And so a lot of the work that Gene Bennett did and Spark really opened them up to using maze tests, things like that, to be able to get a better sense of visual improvement. Again, another one of the things we always teach our residents is that you cannot measure vision. Vision is a subjective concept, right? So that includes so many things: peripheral, contrast, motion, so many things. We can break it down into little tiny bits and try to measure them, but in the end, that doesn't give you the full picture. Only an individual has that. So we never measure someone's vision. We measure your visual acuity, we measure your contrast, things like that. And so it took a while for the FDA to understand that visual acuity is not vision. But they get a little scared because visual acuity is easy to measure, and vision you can't really measure. So there's some gray zone in there, and they're more and more amenable to looking into that, but it still remains difficult to kind of change course.
Yeah, I was just going to add that while at Retina International's meeting last week in Iceland, there were several discussions about it, and they pointed out that a patient can vary from 2 to 5 letters day to day in visual acuity tests. So that has to be worked into, and it kind of gives evidence of it's not a finite, as you say, that it varies. Patients have better vision one day than another, or are concentrating more.
Well, you just gave the argument that the FDA has. So they have such a wide, they want to see such a big change to account for any day-to-day variability, and that's very true. The lower the visual acuity you have, the more people in this room know this. It depends on lighting, right? If you walk into your ophthalmologist's office one day and they happen to have the lights on higher or lower, you may do better or worse. So there's a lot of things that go into that, especially with folks with ultra low or low vision. And so measuring other things and making them objective, that's hard. And that's what the FDA wants ultimately to be able to determine. Is it a real finding, or is it just within the noise of the tests that you're doing? And that's what companies and scientists and researchers, we want to know that too. I mean, if your objectives are correct, you want to help patients. You don't want to just have a number look good on a piece of paper and publish a paper, right? You want to actually be able to make a difference, because people are putting themselves in harm's way. No matter how safe a procedure might be or how routine it may be, it's still a surgery. It's still an intervention, and there's possibilities for things to go wrong or to get worse. And so we don't want to fool ourselves. We don't want to fool patients, and that can be hard to do, even with the best intentions.
Thank you.
Preservation of vision is a very difficult thing to prove, and it's very difficult, and it's very expensive to prove. So if you're a company that wants to develop a therapeutic that's going to slow things down, that may take years and years and years to do that, you need a lot of people. You need to follow them for a long time, and it's millions of dollars to be able to do that. So a lot of times it's not the FDA. The FDA is perfectly fine with preservation of vision and or anatomy and function. But how long do you wait? Because unless you have a lot of patients to follow, how do we know that you wouldn't have been stable anyway? Right. So in order to be able to make that something that you're going to then turn around and ask the insurance companies to pay $400,000, for a treatment for, they want to be sure that that's not just some random effect, that, you would have had that happen anyway. That requires a lot of patients who are treated, untreated, followed at the same time. So when you have a more rare disease, that's hard to do. And it also takes longer to follow them longitudinally. So those are all things that have to go into it, though, and we try to avoid the one-offs because there's plenty of snake oil out there that can find the one patient that said, hey, you rubbed that lotion on the top of my head, I haven't gotten any worse. Right. But we don't want to pay $400,000 for the lotion that you rubbed on top of your head.
When you think about things like adaptive optics, where literally within a year or two, you could tell if a few photoreceptors have died or not, because it is so refined, yet that is not approved yet by the FDA. Or, for instance, Conan has come out recently with a new type of, it's not a subjective perimetry test, microperimetry test. It's objective. I've actually taken the test, and it's amazing where we don't have to find a focal point to stare at, and things are beeping. It's all done for you. You just kind of stare straight ahead.
We're being told we have to get,
it maps your periphery, and it's amazing.
We're being told we have to get ahead with the rest of our talk, but actually, that blew over my head. So that is a great point. So biomarkers. So using these newer technologies to be able to say, okay, at 1 year, I've got enough. Even though you're maybe haven't changed, I've got enough evidence to be able to tell, as opposed to watching you for 5, 6, 7, 8 years. That is a great application of these endpoints so that you can see them a little earlier. So that's a very good point. So using them in that regard, I think is very important, and it is something that they're slow to adapt because so few people. And then again, you go back to adaptive optics. We can talk more about it later, but adaptive optics is not what's the word? It's not uniformly across the board. So everybody's adaptive optics machine is slightly different. It's all made by Alf Dubra and sometimes Jeremy Rogers. Not everybody. It's not everybody that does it is not standardized. That's the word. It's not standardized. So if I do it in 2 different places and they do it 2 different ways. So in many respects, the field has to be able to step up and say, we're going to talk to one another, we're going to standardize things, and then we're going to prove to the FDA that this is a reliable test, that if you get it at Penn or you get it in Madison or you get it in Europe, you're going to get the same results. Right. So you have to do those sorts of things first too, but it's slow, and sometimes it's a matter of funding and things like that too. So, but it's a very good point. I think it'll help these types of therapies when we're looking at preservation of vision as opposed to restoration.